Hi, today we're interviewing Dr. Jennifer Thomson. She's a geneticist from South Africa. She has done a lot of interesting work in the area, and she's currently working on drought-resistant maize. She's actually doing it in 14 different varieties. Good morning.
Good morning.
So, can you tell us a little bit more about this project, how it was born, and why you're using so many different varieties?
Okay, so the genes are derived from a resurrection plant, which is called Xerophyta viscosa. It grows in cracks in rocks in mountainous areas in South Africa, so it's often subjected to great desiccation, also high temperatures, cold, and salinity. So we figured that it must have a lot of interesting genes. So we made gene libraries and looked for genes that were upregulated during dehydration and stress. And we sequenced a lot of them, over nearly 100 genes. And then we blasted them against for homology to see if any of them were physiologically interesting. One of them we chose has no homology to anything in the gene banks. It's a transmembrane protein that sits in the membrane and signals stress to other genes inside. One of them is an codes for an osmoprotectant, sorbitol, so it's like antifreeze, and the other is an antioxidant that mops up oxygen radicals. Uh, we have tested it in the lab in a number of plants, and now we are going into the glasshouse to test them in maize. The reason we're using 14 different varieties is because we don't want to stick with one variety that might not work well in the field. And so what we're doing is actually transforming two more or less readily transformable lines that are inbred, and we'll then cross them out into the 14 different varieties to see which ones grow in different parts of South Africa and the rest of Africa, because this is an Africa-wide project.
So, you're transferring the traits in a few varieties and then using traditional plant breeding.
That's right. We need the plant breeders to cross them into the different lines. Also, the genes we have chosen are also upregulated. They're only induced during stress conditions, so they couldn't form a load or a metabolic load on the plant during normal conditions.
Well, roughly, what will be the cost of development of each one of these varieties?
Well, we've got a grant of South African rands 12.5 million, which translates into just less than 1 million US dollars. And we figured that should be enough to get us to the glasshouse stage. Then we've got to do field testing. And of course, I have no idea what that will cost, but it's
Can you
also.
give us a ballpark estimate of the regulatory cost until one of these varieties makes it to the market?
Well, the South African government is very much in favor of GM crops. We have our maize currently is about between 80 and 90% GM in South Africa, and this grant is from the South African government. So the regulatory problems should be fairly straightforward, but we have to comply with them. So it could take another 5 to 6 years.
Okay. And in our countries, do you have any estimate of the amount of money will take to cover the regulatory, like the feeding test and all those regulations?
Well, it'll be the same in other countries. So, you just have to multiply it by the number of countries you want to test it in.
So we're in a situation where the regulatory costs are actually a lot more than development costs, which are quite modest for 14 different varieties.
Yes. No, that's absolutely true. The regulatory costs are the ones that are killing us, which is why one of my other projects has never seen the light of day, because the problem is a virus that's only found on maize in Africa. And even the multinationals say that with their large coffers of money available to them, they won't recoup the expenses of the regulatory costs because it's only an African problem. Fortunately for us, unfortunately for the rest of the world, drought is a ubiquitous problem. And so if it works in Africa, it should work on other continents as well.
So basically, the size of the market is just too small to support all these regulatory costs.
That's the problem,
So you
yes.
have a situation kinda similar like the neglected diseases, but here you have the solution. You
Right,
just cannot use it. In
exactly.
neglected diseases, we don't
They
have
don't have,
solutions.
yes.
It's even worse.
It's far worse. It's far worse. It's actually criminal to think that the anti-GMO lobby is making it so expensive that poor farmers cannot have the benefit of these technologies. It's criminal.
So if we could have like a small company born in Africa competing against Monsanto, and that is not happening because the regulatory burden is just too high.
Yes, the seed companies in South Africa are just not strong enough financially to be able to handle this regulatory burden.
It's really sad that I know people mean well and they have good intentions, but they only favor the monopoly that they hate so much.
It's true. I mean, if it weren't for the huge expenses that the anti-GMO lobby is imposing, many small companies, many small seed companies could produce these GM crops and make money, and also make crops that are valuable to smallholder farmers. It's it's crazy that the antis are saying, on the one hand, it's the multinationals that are reaping the benefits, when it's their requirements that allow only the multinationals to be able to afford all these regulatory costs.
Okay. Can you tell us a little bit more about your previous project with the virus-resistant corn?
Yes, there is a virus in Africa that affects maize. It is called maize streak virus. It's spread by a leafhopper, and it can wipe out a crop when it infects. Unfortunately, the epidemiology of it is not very clear, so we never can anticipate when the virus is going to strike. But what we do know is that when plants are stressed under dehydration, under drought conditions, they're far more susceptible to this disease. So it can only get worse. So we have, over the period of the last 10 years, developed maize streak virus resistant maize that is hugely resistant. We've got in one crop, in one plant we can put in 3 different approaches. So resistance from the virus's point of view is hard to imagine, but it's just an African problem. So there is not enough money in Africa to make it worthwhile for any company, multinational or small, to pay the costs of development in Africa for a problem that's only in Africa.
And all these approaches that you said you used 3 different approaches. These strategies are specific for the virus in its implementation, but the concept is general, right?
Oh,
You could
yes.
use it for other diseases.
Well, they've used it for cassava mosaic virus. You see, these 2 viruses have DNA as their genetic material. Most plant viruses have RNA. So most resistances, like to the papaya ring spot virus, is using the coat protein. For various reasons, that wouldn't work for DNA viruses. So this approach has been used for the DNA viruses, the so-called Gemini viruses. They have little twin capsids.
So basically, the situation is that 10 years ago you developed this particular application based on general techniques that since then will have been used for a lot of plant pathogens,
Yes,
but no one has done it because
it's too expensive. The regulatory costs are too expensive.
And the final question, what do you think will be the next steps to finally unlock these tools and let people use them? What should we move into?
For instance, animal feeding trials tell us nothing. So a protein is a protein, whether it comes from an inserted gene or the plant's own gene. So animal feeding trials are telling us nothing. We can do allergenicity tests very simply on the skin of a rabbit. You don't have to kill rats to do animal feeding trials to show that a protein is a protein and it's going to be degraded into amino acids. So if that were to be dropped, and if the field trials were to be not have to be fenced and regulated because of potential vandalism from the anti-GMO lobby, then it would become a lot less expensive.
Thank you very much, Dr. Thompson.